Welcome to BioWise with Aanchal and the second part of the chapter, that is structure of chromosomes, cell cycle and cell division. In the first part of the video, you have already seen the different nucleic acids, that is DNA, RNA, the nucleosome model, the structure of DNA, nucleotide. So let us have a very quick revision so that you understand that what we have done earlier. So in this chapter, we have already studied about the nucleotide. Remember, this is your nucleotide and you have the phosphate, okay, and this is the deoxyribose. So this is your nucleotide. The nucleotide sequences is the gene, remember. Now the gene is the unit of inheritance, a specific nucleotide sequences that encode for certain proteins. So we have already studied this. Now what happens to these genes, or these genes are there on the DNA, right? So now the cell division. Cell division. When you talk about the cell division, cell division is what? A cell has to divide, then grow, and then again divide, that is redivide. So this is your cell division. What is the need of cell division? Now children, since you are the students of science, right? So one question should always come to your mind, that is why something happens. If I say cell divides, the question should be why the cell divides. Now cell divides because of certain reasons. What are those? The growth. Then you have repair. The wear and tear of tissues takes place. So it's very much required. For example, you get hurt, you cut yourself, there is a gap in between the skin, it has to heal. So that healing is because of the cell division. Then regeneration. And finally, the reproduction. So cell division helps in all these processes. So that is the need of cell division. Is it clear? You can take a screenshot. May I rub the board? I'll rub it quickly. Now when we talk about cell division, first thing that you should understand that how many chromosomes the cells have. So let us take an example of human beings. So when you talk about a human being, okay, the human being. Now you know that what is the number of chromosomes in human? That is 46, or you can say 23 pairs. So for the body cells, there are two genders, that is male and female, okay. So for the general body cells, the male has 22 pairs of autosomes, and same is the case of the females, 22 pairs of autosomes. And when you talk about allosomes or sex chromosomes, the males have got XY and the female has got XX. Remember? Similarly, when you talk about the gametes, now in gametes, the sperm. What is the composition of sperm? 22 plus X or 22 plus Y. That is, the number of chromosome is halved. Similarly, in the egg, the number of chromosome is 22 plus X. So depending upon which fusion takes place, the male child or the female child is born with the restored number of chromosome, that is 23. I hope this is clear. So now we will move to the next topic, that is cell cycle. Now when you talk about the cell cycle, it has got two phases. The first one is the resting phase or the non-dividing phase, which is called interphase, and the second phase is the dividing phase, that is called the mitotic phase. Dividing phase, that is called the mitotic phase, right. Let me quickly draw the diagram of the cell cycle. Now when you want to understand the cell cycle, I have drawn the rough diagram. Labeling, I will do it in front of you. The first phase of the resting phase, that is interphase. What is resting phase? From here to this phase, that is, this is called the interphase. It is the longest phase of the cell cycle. It has got G1 phase, S phase and G2 phase. What is G1? G1 is first growth phase, S is synthesis phase and G2 is growth 2 phase, followed by this step, which is the mitotic phase. Mitotic phase is what? The dividing phase. After the mitotic phase, you get two cells, okay, and this one cell again enters into the G1 phase. There are certain cells which do not divide, so they slip into another phase, that is called G0 phase, which does not undergo any kind of cell division, okay. They remain in the G0 phase. Now what happens in these phases? I will come to the mitotic phase little later. Now the mitotic phase is the dividing phase, the interphase is the resting phase. In the resting phase, the cell prepares itself to divide. So what happens in the G1 phase? In G1 phase, the mitochondria, the chloroplast divides, and necessary proteins and RNA required for the S phase is synthesized in the G1 phase, okay. Is that clear? So you have RNA, you have proteins, you have mitochondria, you have chloroplast, these all divide in the G1 phase. The synthesis phase is the phase where the DNA replication happens. Children, this is very important, okay, that is the DNA replication. So S phase is the synthesis phase where DNA synthesis takes place, followed by the G2 phase. It is shorter than the G1 phase. G2 phase, the necessary mRNA, that is necessary RNA and proteins are synthesized for the mitotic division, okay. Now the cell is ready to divide. Now everywhere there is a checkpoint which checks whether the required processes have taken place correctly or not. The DNA has replicated correctly or not. So there is always a checkpoint, understood. Now what happens in certain cells that we call cancer cells? These checkpoints are lost or they do not function properly. So the cell enters into an uncontrolled cell division, in which the cell continuously divides and divides and divides, forms a tumor, cancer, benign cancer, malignant cancer and so on, and the problem happens because of the uncontrolled cell division, where the cell could not even mature before dividing, right. That is why it is a disease. So cancer, the checkpoints of the cell division does not function, but in normal cells, if there is any kind of error that is happening, the cell division stops and the cell dies. Is that clear? That is called apoptosis, cell death, not included in your syllabus, but still for your information. Now after the cell is ready to divide, it undergoes the mitotic phase or the M phase. The mitotic phase has got two divisions. One is karyokinesis, that is the nuclear division, followed by cytokinesis, that is your cytoplasmic division. Is that clear? So, now what happens in karyokinesis? This phase is the karyokinesis and followed by you have cytokinesis. Okay. In karyokinesis, what happens? You have prophase, right? First phase is the prophase, followed by metaphase. Metaphase was asked in 2025 examination. Metaphase. Then you have got anaphase, followed by telophase, right? So, this is your different phases of mitosis, karyokinesis. Is that clear? Now, this is what the cell cycle looks like. You can take a screenshot. The diagram of this picture will be in the description. You can download it and use it for your studies. Is this clear, children? Please take a screenshot if you want. Let me rub it quickly. Yes. Now, before going into the mitotic phase, let us first understand what happens in the S phase. Now, in the S phase, generally a cell contains, I am just drawing the nucleus. It contains chromosomes or this is the chromosomes. During S phase, what happens? This is entering into the S phase, the nucleus, the chromosomes will, that is the chromatids will double itself, will duplicate. So, instead of a dyad, that is 2 chromatids, it becomes 4, and this happens in S phase, but they still remain as a double line. They do not just appear as a chromosome. Understood? So, what happens? The chromatin fiber, which is like this, it doubles and it remains in this structure, in this form only. Understood? Now, how this happens is the DNA replication. Now, DNA is a macromolecule, you know that, made up of nucleotides. It has got ATGC bonding. If you remember, I have taught in the first class, you know that A double bonds with T and G triple bonds with C. This is the complementary base pairing rule. Now, when this parental DNA forms the daughter DNA, you have the parental strand being conserved in the daughter's strand. Understood what is happening? So, here you can see this orange line, that is the parental strand. So, the daughter DNA contains one parental strand, okay, one DNA becoming 2. Since they contain half of the parental strand, the entire process is called semi, that is half, conservative, because it is conserving half of the parental strand. So, semi conservative DNA replication. Okay, so our DNA replication is semi conservative in nature, and when this process happens in the S phase of interphase. Mark it, very important. Is that clear? Let me rub the board. Now, next we will be studying about the different phases of mitosis. It is very simple, the diagrams are easy and can be asked for drawing in the examination. So, let us study about mitosis, the different phases. Okay. As you all know that the cell in the interphase is the nucleus, and you have the chromatin fibers and obviously a nucleus and a centriole, right. This is a cell and interphase. Right. Now, after the interphase, the cell enters into the prophase. So, for prophase, how will you recognize whether it's a prophase? First of all, the nuclear membrane will be dotted line. That shows that the nuclear membrane is disappearing, and the chromosomes becomes thick and condensed. I am drawing the diagram for the 4 chromosomes, right, in the daughter cells or in the parental cells. 4 chromosomes, and that is your centrioles will start moving apart from each other, and you have the spindle fiber formation begins. Is okay. Now, this is your prophase. This is your prophase. Is it clear? In prophase, you have got the spindle fibers. That is, spindle fiber formation begins. You have the centriole. You have the nuclear membrane disappearing. Even the nucleolus also disappears. Nuclear membrane disappears, right? And you have chromosomes. This is how you are going to draw and label, and you get your full marks. After prophase, the next phase is metaphase. In metaphase, what happens? The centriole has already moved to the pole, and you have got the spindle fiber formation totally complete, right? Since it's metaphase, you have a equatorial line. There is a middle line, M for metaphase, M for the middle line, right? In the middle line, the chromosomes align themselves on the equatorial plate. So, you can draw this way. Yes, you can draw the chromosomes. I have used 4 chromosomes here also. They are just aligned on the equatorial plate. Followed by metaphase is the anaphase. In anaphase, the chromosomes move away from each other. They drift away. A for anaphase, A for away. So, now the chromosomes move away from each other. Okay, so the chromosomes move away from each other. Okay, the chromosomes move away from each other. This is your anaphase. Anaphase is away. Okay, this is anaphase. After anaphase, that is, we have got what? Prophase, then you have metaphase, then you have got anaphase. After anaphase, the next phase is the telophase. In the telophase, the constriction begins. That is, furrow formation begins. Okay, furrow formation is a unique characteristic for the cell division in animal cells. In plant cells, you have cell plate formation, no furrow formation. Again, in plant cells, there is no centriole. So, the spindle fiber begins from the microtubule present in the cytoplasm. Is it clear? So, in plant cells, the spindle fiber formation is there, but there are no asters. Since we have got the centrioles in the animal cells, asters are also present in animal cells. In plant cells, there are no asters. Okay? And in plant cells, since centrioles are not there, spindle fiber begins from the microtubules present in the cytoplasm. So, spindle fiber just appears in the cytoplasm, unlike the animal cells. So, let us go back to telophase. In telophase, what happens? The nuclear membrane reappears, and the centriole is towards the pole. right? And these chromosomes again, you can see here, they just form the, right, later they will again form the chromatin fibers. So, we started with one, finally we have received one only, right? So, now this is telophase, this is telophase. This process is, what is it termed as? Karyokinesis. This entire process what I have done on the board is karyokinesis, that is nuclear division. After karyokinesis, the next step is cytokinesis. This is cytokinesis. You have two daughter cells formed, right? Two daughter cells with 4 chromosomes, and again they will go back into the chromatin fiber state. The nucleolus will reappear. So, you have got 2 daughter cells. They have got 2 daughter cells. And what is the number of chromosomes? We started with the 4 chromosomes. Here also you have 4 chromosomes. So, here the cell which was in 2n condition give rise to 2 2n condition daughter cells. Is that clear? You can take a screenshot if you want. The diagram again will be put into the description. You can download it and use it for your study purpose. So, I will rub the board quickly. Yes, I hope you understood about mitosis and the different phases of mitosis. Now, there is a difference between mitosis in plant cells and animal cells. As I was drawing the diagram, that time I told you that in plant cells we have centrioles as a unique structure, and plant cells do not have centrioles. So, now if plant cells do not have centrioles, then what helps in the synthesis of spindle fibers? I have told you the answer. Let me see who all remember it. Put the answer in the comment section. How the spindle fiber is formed in the plant cells. Okay. Now, another thing is aster formation does not take place in the plant cells because plant cells lack centrioles. In animal cells, you have aster formation. In animal cells, cytokinesis happens by furrowing. In plant cells, cytokinesis happens by cell plate formation. In plant cells, you will have cell wall and the cell membrane, you can easily identify, and there will be no centrioles, right. The question can be given in the exam where they can ask you, identify the cell as plant or animal and justify your answer. Now, if you see there is a centriole present, kindly make it an animal cell, and if there are 2 membranes, one cell wall, one cell membrane, and there is no centriole, it has to be a plant cell. In plant cell, cell division by cell plate formation. In animal cell by furrow formation. I hope mitosis is clear. What is the significance of mitosis? There are 4 significance. G, R, R, R. Growth, repair, regeneration and reproduction. Asexual reproduction generally happens in mitosis. I hope mitosis is clear. Then is the next phase called meiosis. Now, meiosis is not there in your syllabus. In meiosis, you have to remember there are 2 steps that happen in meiosis. Meiosis 1 and meiosis 2. Meiosis 1 is a reduction division in which the chromosome number is reduced to half. If you consider human, 46 becomes 23. This is the reduction division. Fine. In meiosis 2, it is similar to the mitotic division. So, what happens? These 2 cells will again form 23 and 23, and this 23 will again form 23 and 23. So, the final outcome of meiosis is 4 haploid daughter cells. Okay, we started with a 2n condition, that is diploid condition, and we ended up into 4 haploid condition, 2n, and this is n. I hope it is clear. What is the significance of meiosis? It gives rise to variations, right? Then it also helps in restoration of the chromosome number. Since gametes formed will have N condition. So, two N's, that is male and the female gamete, when they combine they form 2N. So, the chromosome number is restored because of meiosis. Now, I have said that meiosis is the cause of variation. For that we have to study the last part of the chapter, that is synapsis. Is this clear? Now, what is synapsis? Synapsis, you'll hear a similar word when we study nervous system, synapse, that is the junction of neurons. Synapsis, sister. So, synapsis happens in the cell division chapter. So, in this what happens, let me use the traditional colors to make homologous chromosomes. So, if you have a homologous chromosome, right, this is from parent father and this chromosome is from the mother. Okay. Now, these two are the homologous chromosomes. What are homologous chromosomes? The chromosomes which are having same shape, size, structure and is inherited from either of the parents, the homologous chromosomes. These are chromatids, sister chromatids, and this is sister chromatids. Okay, and these two are the non-sister chromatids. They are the non-sister chromatids. Okay, crossing over takes place between the non-sister chromatids. Now, what happens? You can see this chromosome and the other one will have a X-shaped formation. Okay, this X-shaped formation is called chiasma. This X shape is called chiasma, and after the chiasma formation there is a exchange of genetic material. There is an exchange of genetic material that will take place. Okay, so this will become blue and this part will become pink. So, this will give rise to a variation, and this process of exchange of genetic material is called crossing over. Is this clear? What is synapsis, what is chiasma and what is crossing over? If I have to make you understand in a simple example, suppose there are two friends. When they come to meet each other, coming closer to each other, meeting each other is synapsis. Then they shake hands. So, taking a photograph of shaking hand, that is the evidence that they have crossed each other or they have met each other is the chiasma formation. And if they exchange gifts, it is called crossing over. Understood? So, meeting of two friends is synapsis, shaking hands and taking a photograph of it is chiasma, and exchange of gift is called crossing over. Is it clear? Let me rub the board. Now, the last point of this chapter is the difference between mitosis and meiosis. So, how will you differentiate? Now, in which cells does the mitosis occur? It occurs in the somatic cells, and meiosis occurs in the reproductive cells. Number of daughter cells formed. Mitosis, there is how many? Write in the comment section. How many daughter cells are formed in mitosis and how many are formed in the meiosis? The answer must be given in the comment section. I have already told you before. Right, mitosis can happen throughout the lifetime. Whenever you need any cell repair or you can say growth. So, mitosis happens all the time. Meiosis happens only during gametogenesis. What is gametogenesis? Formation of gametes. Right, gametogenesis is formation of gametes. What are gametes? That is spermatogenesis and oogenesis. spermatogenesis, and you have oogenesis. So, during this phase only the meiosis occurs. Okay. So, the number of chromosomes in the gametes are halved, so that when they undergo the process of fertilization, the chromosome number of the species is restored. So, this is the entire chapter. I think I have explained it properly to you all. If you have any doubt regarding any topic that you have not understood, put it in the comment section, it will be answered. Happy learning, students. Have a great day.